I'm Mohammed Kashkush.
I'm David Katz.
And I'm Manu Patel. We are student pharmacists at the University of Pittsburgh, and today we're going to tell you about why we're sending bacteria and antibiotics into space. When bacteria infect our bodies, we use antibiotics to help our immune system kill them. Like humans, bacteria will do anything to survive, like mutating to prevent antibiotics from working. When this happens, we say that bacteria become antibiotic resistant or antibiotics no longer work for them, leaving patients with fewer treatment options. Today, over 700,000 people are killed per year from resistant bacteria, and this problem is only getting worse. By 2050, over 10 million people will be killed by resistant bacteria.
In space, antibiotic resistance is more dangerous than it is on Earth. Confined spacecrafts make it easier for infections to spread from person to person, and our immune systems are weakened in space, making it harder for our body to clear the infection. And in microgravity conditions, the bacteria are more likely to develop resistance. In fact, in the 1960s, a cosmonaut on a Soviet space station was treated with an antibiotic. But unfortunately, some resistant bacteria survived and infected another cosmonaut. But he couldn't be treated with the same antibiotic. So to outsmart bacteria with new medical strategies, we will look into how they react when threatened with an antibiotic. This leads to our research question. How does microgravity affect E. coli response to ciprofloxacin? Answering our question will provide insight into how E. coli adapts in these conditions, and our results will identify ways to prevent antibiotic resistance in space and on Earth.
So believe it or not, there's actually more bacteria cells in your own bodies than there are human cells. As long as these symbiotic bacteria follow the rules, they're actually quite healthy and don't cause any disease. One of the bacteria that make up your gut microbiome is E. coli. But if E. coli finds its way out of your gut, it can actually cause infections, and we usually use an antibiotic called ciprofloxacin to treat that. We also like to call it Cipro, just for short. But to get inside the E. coli and reach its intracellular targets, Cipro actually has to cross first, has to cross two cell membranes and the cell wall, move through the cytoplasm all the way towards the DNA, and only at that point can it bind to two enzymes that are required for cellular replication. And if all of that is successful, then ultimately it'll prevent E. coli from splitting from one bacteria into two, and ultimately the population will die.
However, E. coli will try to stop Cipro at multiple points through resistant mechanisms, like pumping Cipro out of its cell, changing its own targets, or upregulating its genes to prevent Cipro from entering. This can all be measured through E. coli's transcriptome, or collection of RNA. This is how our project uses the power of transcriptomics to answer our research question.
5 days before Mission 13 payload returns to Earth, our experiment will begin. This is when the astronaut will remove the first clamp, which will mix the E. coli inoculum with the growth media that contains the antibiotic Cipro.
Since there will be a mix of Cipro, E. coli, and growth media, we needed to make sure that the bacteria would survive in this mix, so we performed tests in the lab. If too much Cipro is used, then not enough bacteria will survive for analysis. If too little is used, then the bacteria will hardly be challenged to respond with altered gene expression.
3 days later, our experiment will end. This is when the astronaut will remove the second clamp, which will add DNA-RNA shield into the mix. DNA-RNA shield will stop all growth, and it will also bind to the RNA, preserving this RNA for 30 days at room temperature, which makes this product ideal for our experiment's time frame and temperature constraints.
So the mini laboratory will remain in that state until it returns back to our hands in Pittsburgh about a month later. So now that we have a final sample containing pure E. coli RNA, we can use transcriptomics to determine the exact identities and quantities of the RNA molecules. However, that kind of information is really only useful if you already know beforehand what role each unique RNA molecule plays in the bacteria. So that's why we selected a specific strain of E. coli that had a fully mapped genome from the scientific community. With a fully mapped genome and RNA data, what you can do is you can actually create a differential gene expression profile from our analysis between the spaceflight samples and the ground truth samples. And this allows us to compare what functions and thus what genes were involved in the survival response to ciprofloxacin. So for example, if the RNA levels are significantly different between the two environments, you know that the gene and thus the function associated with the gene probably plays an important role in the survival response. In contrast, if the RNA levels are similar, the gene associated with the RNA of interest is it's likely not affected by a change in gravity.
Here are the references we used. And we'd also like to thank all of our mentors for their support, sponsorship, and advice to make this viable idea into an experiment.
Before we finish, imagine what the future will look like. We believe that Homo sapiens will be a multiplanetary species. This isn't so far-fetched, considering commercial flights to the International Space Station are already being offered today. And just like on Earth, these societies will need doctors, pharmacists, and scientists to understand how to treat people in outer space. Working towards this future is the ultimate motivation behind our research. And who knows, if the scientific community is successful, one day, any one of you may have a ticket to Mars with your name on it. Thank you very much.
Do we have any questions for this team?
Hello, I'm Rubens from New York City. I would like to know what was the strategy to calculate the balance between the E. coli and Cipro?
Yeah, so in the lab, our mentor Christian and us, we worked to see how much bacteria would be at the end of certain concentrations of Cipro we used. So we started off with the minimum inhibitory concentration, which is the concentration we ended up going with. And then we went a few dilutions above and below. And we found that the minimum inhibitory concentration will give us the ideal amount for analysis.
Also, even just to add to that, we also had the pleasure of working with people all the way in Colorado who had a lot of experience sending the same strain of E. coli up to the space station. And they shared with us some of their data about how this strain of E. coli grew in space, and it gave us some more background information that was helpful.
Hi, I'm Mike from Galloway, New Jersey. Just curious if you guys have a prediction about what you might see comparing your space flight to your ground control.
So we've looked into past literature and past experiments. Like Mo said, we worked with a Colorado professor, and they showed that there was an upregulation of the stress response genes that were found, so we hope to see something similar like that. But in general, it's kind of like a hypothesis generating experiment, so it's just like whatever we see. Yeah.
Thank you.
Yeah.
And come check out our poster. There's a QR code, you can learn a lot more about it. And we really appreciate everyone coming here today. I love the experience.
Hi, I'm Jane from Florida. I'm familiar with Cipro, unfortunately, to the point where I had too much Cipro and I ended up with CITIF. So my question to you guys is, are you taking that into consideration when you're putting this experiment in place? I mean, to the point where you can't use Cipro to the point you're going to eliminate whatever is the body needing the Cipro for, but not enough with CITIF?
Yeah, did you want to go into that? We were talking about this like last week.
Or I don't remember exactly what we talked about, but unfortunately for this experiment, because it's in a test tube and we can't, at least with our design, we only have one bacteria growing in there. And the problem with C. diff, for those of you who don't know, is a lot of times when you take an antibiotic, what happens is the gut microbiome I was referring to earlier, there's actually thousands of bacteria, or strains and different species residing in your gut, and they help you digest foods and things like that. But if you take an antibiotic, you might unintentionally kill the healthy bacteria, allowing dangerous bacteria to overgrow. So this is a serious problem in clinical practice, but for a test tube experiment, we didn't really factor that in.
My name is Matt from Galloway, New Jersey. I was wondering why you guys were going to be starting the experiment toward the later end of the flight and not the beginning. I was wondering if there was like any increased radiation that the E. coli might be affected by, if it would be beneficial to start earlier or later, what your mindset was behind that.
Yeah, so pretty much talking about the time frame constraints on that one. The opportunities that we had were at the beginning of the experiment, we could do a 2-day difference. At the middle, it was a 5-day difference, and then a 3-day difference right at the end. And the growth curve of bacteria, we had to figure out pretty much which time frame really was perfect for ours. And so we picked the last one, and also the preservative that we used could only keep that RNA, which is super unstable, for so long. And for the time frame it takes to come back Earth and send it to us back at Pittsburgh, that ending period was where we picked.
Thank you.
Thank you very much, Pittsburgh. Thank you.